Hello, my name is Carina Immer, and I just finished my PhD at the BMRZ at the Goethe University in Frankfurt. And I was interested in the structure and function of RNA binding proteins and studied them with NMR spectroscopy. So I have been using NMR since my master thesis that I started 5 years ago, and I was using NMR in my PhD. And I just came to the NMR when I moved to Frankfurt for my master. So before I haven't heard about NMR because I come from biology. And in the first lecture about NMR, I got really fascinated and interested in this very complicated method at the first place. And when I started to work with NMR, I really like that you can see the molecules in such great detail. And I think if you once have seen that, it's impossible to go back from this. I think the most powerful thing that NMR can do is that one can get information on the structure of a molecule. For example, I'm working on proteins, so if I just record one spectrum, I can see if the protein is folded. And I like that you can also investigate not only the structure, but also dynamics or interactions with other molecules in great detail. For example, I work on RNA binding proteins, so I can see where the RNA is binding on the protein with NMR. So that really helps me to understand how the protein is functioning and where the RNA is interacting with the protein. So I'm working on proteins, which are important components of every cell, and they have a specific function. And I'm interested in proteins that interact with other molecules in the cell, which is RNA. And I'm now interested in how exactly the protein does look like and which parts of these proteins are important to fulfill its function, because it's very specific for every protein that you can find in the cell. And this function is defined by the structure, so by how it is look like in a three-dimensional way. And this then can help to understand what the protein is doing. We also use biochemical methods in our lab to study, for example, the binding of the protein to the RNA, so to say. So, for example, the binding characteristics, and these results we can then also see in the NMR experiments, where we would then have a bigger resolution of this interaction that can then add up on the complete picture of this interaction.